Okay, dear participants and respected sir, Pushpendra sir, we will be starting our session of today's Science Dialogue series, where we will be having our speakers of today's session, who is Dr. Pushpendra Singh. He will be delivering a talk regarding the evolutionary history of leprosy, Bacilli, and its global dissemination with the human migrations. So before introducing you all guys to our today's speaker, I would like to give a just brief introduction about this Science Dialogue series to everyone. So this Science Dialogue series, it's a brainchild from Dr. Gopal sir, who is, because of certain predefined commitment, he is not able to join us today for this session. So I, on behalf of him, also welcome to everyone, including our today's speaker, Dr. Pushpendra Singh. So, regarding this Science Dialogue series, this series was initiated almost 1.5 years back with a single motto to disseminate the knowledge from the different backgrounds for the students, so that they may be easily getting to interact to get the knowledge from the different expert speakers and the speakers who have joined us in during the Science of Dialogue series. They were from different parts of the country with the with varying specialization in their specific field. So for me, this is a kind of a success for the motto by which this Science Dialogue series is being initiated. So now it's my time for introducing Dr. Pushpendra Singh to all our participants. So Dr. Pushpendra Singh, who is our today's speaker, he is currently acting as a scientist F at the ICMR National Institute of Research in Tribal Health, Jabalpur, Madhya Pradesh. So, sir, he is working on the genomics and molecular epidemiology of the microbial pathogens which are causing leprosy, TB, scrub typhus, and viral diseases. Dr. Singh has done his PhD from the National Jalma Institute for the Leprosy and Other Microbacterial Diseases in 2008 with a specialization in mycobacterium. He pursued a postdoctoral fellowship at EPFL Lausanne, Switzerland. Later on, he joined MSU Baroda at the Department of Biotechnology in 2015 to 2016. That was the time I actually have a very short period of interacting time with Dr. Pushpendra Singh. So later on, he moved to ICMR-NIRTH, where he is currently functioning as a scientist F. The major focus of Dr. Pushpendra Singh is on communicable diseases. His research work has been awarded on the multiple platforms. He has published more than 100 articles and more than 4,000 citations, and H index of 40 is itself a kind of uh, credential for his caliber in the field of communicable diseases. His major research work has been published in Science, Nature, Genetics, and Journal of Immunology, which are very few to name, and many others more journal articles are there. I think that now though there are many accolades that I can count upon Dr. Pushpendra Singh, but this much is sufficiently enough to make you all guys aware that what kind of research caliber Dr. Pushpendra Singh is holding. So, sir, I welcome you, and I definitely believe that our participants who are from the BSc or MSc or even postdoc, even the PhD scholars, even some faculties, they have also joined us. So all of them will be getting benefited from your vast experience and your interest area of this communicable diseases. So, sir, I welcome you on behalf of this Science Dialogue series. So I just now hand over this virtual platform to you so that we can start the session of today's.
Thank you, madam, for the kind introduction and for giving this opportunity. I also thank Dr. Gopal sir for keeping this series alive and regularly hosting such a valuable interaction between different participants and different speakers from different parts of the country and abroad also. So I'm really thankful to be able to connect with you all and thankful for this opportunity to share our work in last more than 1 decade that was conducted in different laboratories in Switzerland and USA. And after return from USA, I have conducted part of this work at MSU Baroda. And currently, I'm at ICMR National Institute of Research in Tribal Health at Jabalpur, which is the geographical, very close to the geographical center of the India. Um, it's right in the heart of the country, basically. And I hope you are able to see the presentation. I'm putting that in the full screen mode. So today I would be basically giving an overview about the evolutionary history of leprosy, bacilli, and its global dissemination with human migrations. So as I briefly mentioned, part of this work was conducted at Switzerland, at Global Health Institute in Lausanne, which is very close to Geneva. And at the time, we were also collaborating with Better National Hansen's Disease Program in United States. I later on moved there for 2 years, and with Ramalinga Science Fellowship, I joined at MS University of Baroda in 2016, and from there, in 2019, I joined ICMR as a scientist E, and since then I have been working there, and recently have joined as scientist F. So, giving a brief overview about our institute. Earlier, it was established as a regional medical research center for tribals, or briefly RMRCT, in 1984, and considering its national level contributions and mandate, it was renamed as National Institute of Research in Tribal Health in 2014. And this is the only institute which is solely dedicated for health research in tribes. It has a mandate of identification, estimation, and comprehensive care of communicable and non-communicable diseases prevalent in tribal communities across the country. It also provides technical support for local, state, and regional health functionaries, and also provides policy-related guidance and evidence to the government in planning, monitoring, and evaluation of regional health programs. We also provide diagnostic services for all population uh, under our in the areas where we are working through our main campus as well as through our various field centers and the rural health research units in Madhya Pradesh and Chhattisgarh. So it started with a very small beginning at the medical college, which is in Jabalpur, very close to our current campus, and it has since then expanded itself into the current campus, and now we are providing um, various field programs in different states, including um, entire India actually, through various projects. And in Madhya Pradesh and Chhattisgarh also, we are conducting several door-to-door visits or population-based surveys, where particularly among the tribal population regarding their nutrition, tuberculosis. For example, one of our study that I was also part of included this type of handheld X-rays, where digital X-ray can be obtained at the doorstep of the suspected TB patients, and it can be remotely examined by a radiologist, and diagnosis of TB can be done, including handheld X-ray as well as molecular diagnostics, such as TrueNAT machine, as shown here. So, in addition to these field surveys, we are also providing various molecular diagnostics, and these studies have been useful for molecular epidemiology and drug resistance determination. So, coming to the main theme of the today's talk, it's about the leprosy. And as you may know, that leprosy, or in Hindi we call it Kusht, it basically has a very ancient uh, uh, occurrence in Indian civilization, because it's mentioned in Sushrut Samhita from 600 year before the Common Era. So, because of that, It was believed that leprosy originated in India, but it later on was found to be wrong, and we will see that in our subsequent slides. Uh, Despite the common belief that leprosy is a disease of past, it's surprising that nearly 200,000 cases are recorded globally each year, and more than half of them are in India. It causes deformities if not diagnosed timely because it leads to the loss of sensation in peripheral nerves, and that leads to secondary infection, and gradually it can cause irreversible deformities or loss of limbs as shown in this picture. But it's important to note that leprosy is a very curable disease if diagnosed timely, and free treatment is made available through government uh, by the WHO program, and that's the multi-drug therapy, rifampicin, dapsone, and clofazimine. We know a lot about leprosy bacterium, which is Mycobacterium leprae, but we do not understand its transmission mechanism fully. The most commonly believed mechanism is through aerosol transmission, like for tuberculosis, because these bacteria are also present in nasal mucosa of the infected person, and through coughing, sneezing, it can come into the air, and if somebody gets exposed for a very long period of time, the chances of getting infected are high. However, 95% of the people will never develop disease despite getting exposed uh, because of some natural resistance among them. Few 5% of those people who develop the disease, they take more than 5 to 7 years to develop the symptoms, and that's makes it very. Uh, so that's why it's considered as one of the least infectious diseases. However, it's also a very misunderstood disease. People still have social stigma, and they consider this as a uh, result of some or sin, or sometimes people are also outcasted from by their family, so that's basically very unfortunate, and that leads to hiding the disease by people who may have some initial symptoms, and that leads to the continual transmission. So it's important to note that it's a disease like any other bacterial disease, and it's very curable disease for which people should be not afraid of seeking treatment or guidance. Besides this very slow incubation period of the disease, it has very low genetic diversity, meaning the bacteria present in different people are very much similar, making it difficult to differentiate them. And the molecular epidemiological studies are therefore very challenging for leprosy bacterium. It also remains uncultivated, meaning for tuberculosis, you can culture it and see whether it is growing in presence of drug or not. But for leprosy, this is not possible because its reductive evolution of the this bacterium has led to loss of lot of important genes for its biosynthetic pathways, and probably that is the reason that it doesn't grow in artificial medium. Its genome size has reduced drastically. More than 25% of the genome size has reduced compared to its other closely related mycobacterial species such as Mycobacterium tuberculosis, and it has very few genes which are functional, but all the ones which are necessary for its pathogenesis have been retained by this bacterium. However, due to the reductive evolution, its generation time has increased to 14 days, meaning if somebody has an artificial culture medium, it will take more than 1 year for the appearance of the first colony of Bacillium leprae if one successfully grows it in a lab conditions. Whereas in comparison to other bacteria, for example, E. coli, it divides every 20 minutes. So this is what is the comparison, that 20 minutes compared to 14 days for leprosy. So few examples of application of genomics in leprosy and other infectious diseases. As I mentioned that it doesn't grow in artificial medium. That's why genomics is the only way to study this bacterium. Uh, the PCR-based molecular diagnostics is routinely used for its detection, particularly in the cases where microscopy is negative. Uh, and PCR is more sensitive. Besides, it can also be targeted for the genomic regions which are present in multiple copies in leprosy bacterium. So this approach is very useful for its diagnosis, and for molecular epidemiology also PCR-based studies are carried out. So as I was mentioning that some of the some of the sequences are present in multiple copies. For example, RLab sequence is present in 37 copies. Similarly, so using such multi-copy sequences, it is possible to detect it in different animals also. For example, armadillos are known to have leprosy bacterium in them for several years now. For more than 4 decades, it is known that leprosy is present naturally in armadillos. And it was not clear whether there is a linkage with human leprosy in the United States and the armadillos present there. So we will also see some of the studies which established that link, that as leprosy is a zoonosis in southern United States because of these armadillos having natural infection of leprosy, and people in, in their contact or exposure can get disease from infected armadillos. So we will see those studies in subsequent slides. Besides these red squirrel animals in England and Scotland, they were shown to have characteristic symptoms of leprosy, such as this type of deformities in their eyelid or ear pinna. And it was very surprising that two species of leprosy bacterium were present in these animals, in red squirrels. And recently in Guinea-Bissau National Park, it was found that leprosy was, these pictures showed some disease, uh, skin disease, and these animals were found to have leprosy bacterium. So these chimpanzees were found to be naturally infected with leprosy in Africa, in West Africa. So all these studies are actually conducted in countries where leprosy is not so common compared to India. So we have not actually conducted these well-designed studies in India, whether there are animal reservoirs present, whether they cause any uh, concern to human leprosy also. Are there leprosy bacteria present in soil and environment, water reservoirs, or in some animals? Those studies are being conducted uh, currently to our collaborator, and I'm part of those studies, and we are aiming to investigate that using uh, advanced molecular diagnostic technologies. Besides that, I was mentioning that leprosy, more than half of the leprosy burden is present in India. But when we see the genome information about leprosy strains present in India, we see very limited information. For example, out of the nearly 300 genomes available, only three are currently reported from India. Less than 1% of the strains are represented from India, which account for more than 50% of the leprosy cases around the world. So that we have tried to address, and currently we have sequenced nearly 30 genomes from India using different approaches to remove human DNA from the biopsies so that sufficient coverage of leprosy bacterium can be obtained. And using this approach, we were able to sequence uh, different strains from central India, and we found that they were very similar to each other. They were all of a particular genotype called 1D, SNP type 1D, based on single nucleotide polymorphisms. It was found that they all belong to genotype 1D. But how these genotypes are identified? Uh, a representation of the newly sequenced genomes are shown in this gray highlight, and they cluster together, but some of them cluster with other strains from Southeast Asia or from other countries also, such as uh, Vietnam. So what information we get from this type of genomic comparisons? That is actually very important, because not every sample can be taken for the whole genome sequencing, because it's very expensive also to remove human DNA first and then get enough coverage of leprosy bacterium. But by comparing few available genomes, the power of genomics is to not make it complicated, but to simplify it. Once we compare genomes, we see a trend. For example, some of the SNPs are present only in particular genotype. For example, 1A genotype has some uh, positions which are not present in all remaining genotypes, which are known as from 1A to 4P. And then if you see some more genomic markers which are present in two groups but not in remaining 17 groups. Like that, you have an arrangement of SNPs when you analyze the available data by comparing the whole genomes. And that can make it very easy to identify the genotype by just looking at one or two SNPs. Uh, and we have identified such genomic markers and made it uh, in form of a genotyping scheme, as a revised genotyping scheme, where you have to just see three positions first, uh, as shown in this table in the first three columns. And if you see one of these combination, then you would know what SNP type it is. If it is a GAC, that means it is a SNP type 1. If G has converted into T and that combination changes to TAC, and then the genotype is 2. And to further differentiate, and like that, likewise, genotype 3 and 4 can also be differentiated. Once we have determined the genotype, we can directly determine its further differentiation using few more SNPs. And this is how basically this genotyping scheme can help us determine what genotypes are present. And using this genotyping scheme, it was determined that the leprosy strains present in India are very different from uh, the ones which are reported from Europe. For example, as I was mentioning, that earlier belief was that leprosy originated in India and through Alexander's army it went to Europe. So that was the common belief. But it was found that the genotypes present in Europe are very different from the ones which are in India. And that proved that the leprosy didn't actually reach from India to Europe, but it was through China that the through the Silk Road that uh, strains from Europe reached to China and plague in the reverse direction. So that basically proved some of the previously held beliefs wrong, that leprosy was uh, always present in India, and it was through India that the rest of the world got leprosy. So molecular evidence actually help us clear some of these hypotheses or wrong beliefs, which are actually not true based on these type of genomic evidence. And indeed, it was found that the European strains went to America, as shown in this pink color arrows, through colonization, because the strains which are these uh, pink color strains, which are genotype 3, they reached to America through colonization. And then it spread to the rest of the parts of the South American countries also. And then some of the strains from West Africa, which are known as genotype 4 and are indicated in green color here. Let me use a pointer. So as you can see here, this laser pointer is indicating these green color strains, which are genotype 4. They reached to New World by colonization and slave trade. But this doesn't prove how leprosy, some of the strains of leprosy are not present in some part of the world, and all this indicates about Mycobacterium leprae only, which is the very well-known causative agent of leprosy. But in recent few years, it was indicated that another species called Mycobacterium lepromatosis is present in Mexico and other Caribbean countries in this region. How this actually reached there, it was not clear. If because all the strains from New World were very much similar to European strains, but this species of Mycobacterium lepromatosis that was only present in Mexico and some adjoining countries. When this lepromatosis was found in European skeletons, sorry, in the red squirrels in England, it was very surprising that in a country where there is no leprosy, it has two different causative species in red squirrels. So there was no explanation for that observation. But one unpublished study of one of my colleague, Dr. Charlotte, has recently provided evidence that Mycobacterium lepromatosis was always present in American countries, in South American countries, where lepromatosis was always there, uh, even before the European colonization. Some around 800 to 900 years ago, they found that in one of the database, they found lepromatosis sequences. That study had actually sequenced a human DNA from skeleton sample, and along with in that data, when they analyzed, it was also found to have lepromatosis from the bone samples, because leprosy is known to affect bones. And using that evidence, they showed that leprosy was present in America or South American countries even before European colonization. And it is in the reverse direction that through the colonization, that leprosy bacterium went from Europe to America, but Mycobacterium lepromatosis took the reverse direction. It went to Europe around the same time, and since then it has actually been in red squirrels also. So this is actually very recent evidence. It's not yet published. I had seen these results very recently, and it's under communication. So we hope that it will be out in few weeks in a very reputed journal. So I'm just basically, I would like to give some value of these genomic investigations. Once we know that these SNPs are present, we can develop some simple tests. For example, if we know that this particular SNP is present in a particular area or particular genotype, it can be checked by some restriction enzyme. For example, this restriction enzyme cuts the strains of Indian origin or 1D genotype in this 172 base pair band can be restricted into 172 base pair bands. if that particular SNP is present, and that can differentiate genotype 1D from rest of the genotypes. And this finding was recently published by my PhD student, Ms. Purna Trivedi. Likewise, we are also developing point-of-care diagnostic test for leprosy, where this recombinase polymerase assay can visually differentiate a positive test versus a negative test. Up to 10 bacilli, it can give a very differentiable result. But up to 1 bacilli, that becomes difficult to see on the visually. But on the agarose gel, one can see that there is a band. To make this detection even more simple without needing any agarose gel electrophoresis, uh, one of DBT Biocare awardee scientist, Dr. Shikha Nag, is carrying out a project in our laboratory where a simple lateral flow test can differentiate whether a person has leprosy or not. For example, a positive test would have a line uh, here, and a negative result would be indicating a control line uh, at a different position. And this assay can differentiate leprosy and other microbacterial species also. It has very high sensitivity and specificity because it uses the CRISPR based signal amplification step. So that work is still ongoing. I thank all the funding agencies and collaborators which have supported our work in last 8 years since the return to India, and the other funding agencies in Europe where I was carrying out these studies. So I would just sum up that this study also highlights One Health approach, that if there is a disease agent present in animals or environment, it can cause disease to humans also, as shown here. People have used armadillos for making souvenirs or for as a pet also, or even as a food item, or sometimes accidental exposure as a roadkill animals. So that can actually cause human infection. And when we had analyzed this evidence by whole genome sequencing, we found that the strains in armadillos were exactly identical to the strains in humans. So basically, this finding was the first evidence of genetic involvement of leprosy in United States, but such type of studies are important to be conducted in other parts of the world also. So this was published in New England Journal of Medicine, and the real impact of the study is that when people start to change their behavior for their own safety. After this publication was out, it was covered by media and it was on the Facebook and other social media platforms also, and people started posting that they would stop eating armadillos or they would avoid exposure to armadillos. So that type of change in behavior is actually very encouraging, that people themselves, this type of evidence make awareness among people. And as I was mentioning that leprosy causes skeletal damage. uh, for example, this type of loss of uh, nasal aperture can be detected in skeleton samples also. And using this type of osteological evidences of leprosy, people select such samples for molecular investigation, and using the whole genome sequencing technology, by removing the unwanted DNA and capturing only the targeted DNA on a this type of glass slide, an array slide. This can actually lead to better uh, insights about the strains which were present in past. And using this approach only, we have carried out most of these studies. And as I was mentioning that if there is a damage in uh, during leprosy, this can actually be tracked to the skeleton samples also. Basically, this is what we were investigating. And at that time, we had found Mycobacterium lepromatosis in one of the Mexican patient, and that was also later on described in this type of red squirrels. And now we know how these red squirrels got leprosy, because now we know that it was it came as a reverse direction of colonization. The M. leprae strains went from Europe to America, but Mycobacterium lepromatosis, that's the another causative agent of leprosy, came from America to Europe. So this is basically uh, summary of the current presentation, that natural infection of leprosy is a zoonosis in southern United States that causing local transmission, and current strains of leprosy reached America via colonization from Europe. But now we also know that another species of leprosy causing bacterium came from America to Europe in the reverse direction. And disease agent can remain present in wild animals for centuries undetected. For example, Mycobacterium leprae and Mycobacterium lepromatosis in different animals such as armadillos, red squirrels, and chimpanzees. So, One Health approaches and genomic investigations are very important. Uh, with that, I thank you for your attention, and I would be very happy to take your questions.